Hi everyone, I'm Joe. Pleasure to meet you all. I'll meet you all at some point. I run the Cell Biology Practical. For those of you that were lucky enough to have been in the Cell Biology Practical today, you will know that I am a massive microscope geek. I love microscopes. So this talk today is going to be a bit more tool-based than biology-based, but there's obviously a solid element of biology in there. And when they asked if I had anything I could present, I thought, well, I don't really do electron microscopy and I don't really work on symbiosis, but I'll give a talk about electron microscopy and symbiosis. And the reason for that was because of this guy here, Phil Paul, who's a professor at Oxford. He gave a talk here 4 or 5 years ago, I can't remember exactly when, but it was a fantastic talk. He can talk about symbiosis infinitely, but he can talk infinitely better than I can, but he can talk about symbiosis much more better than I can, so it's really not my area of expertise. I came from the microscopy side of things. The reason why I decided to talk to you about this was because this project was born at a Gatsby summer school 4 or 5 years ago. Phil and I were sat out on the sofas over there, and then we went for a beer, and he asked me a really simple question, and we got chatting, and the question was, could you work out the volume of a single bacteroid, okay? And I said, maybe, and we'll see whether we can, okay? So, lots of work has been done on nitrogen symbiosis. So to grow plants, we use a hell of a lot of fertilizer. It's really bad for the environment. There's some plants, I'm giving a real whistle-stop tour here, okay? There's a lot more on this. There's some plants that naturally can take in symbiotic bacteria and they can fix nitrogen that way. They produce these nodules and it's a real potential tool for us to help agriculture. There's a lot of really fantastic research going on in it, and I'm purely just a microscope guy who's dabbling. So, you know, go and listen to Phil's talk and do other things. But here we've got an image. So this is an electron microscopy image. This is a transmission electron microscopy image of 2 different type of, so this is from P, this is Bacteroids, so A34, and this is from Bean and Bacteroids 4292. And they look a bit different, but they also look quite similar, I would say. Okay, so, you know, roughly maybe the same size, maybe these are a little bit bigger. These have got some kind of striations here, but are these the cells dividing? What's going on here? You know, but we can't be really conclusive. So when we talk about transmission electron microscopy, what we're doing here is we're taking a plant, we're chemically fixing it, freezing it in time, fixing everything within it, and then what we're doing is we are embedding it in a resin, we're making really thin slices of it, and we're taking those thin slices, putting them on a grid, and blasting electrons through it, okay? So I'm a light microscopist, not an electron microscopist, but here's where I dabble in electron microscopy. So traditional TEM is really useful for doing this kind of thing, but is it telling us the whole story? I wouldn't be saying that if it wasn't, would I? So, if we want to think about things in organisation of different bacteria, and can we tell differences between different bacteria, a 2D section isn't really giving us all the information we need to know. What would be amazing is if we could start to look at things in 3D, okay? It breaks my heart to say this. A light microscope can't do that. It's too small. The resolution's far too small, okay? So the resolution of a light microscope, you know, even with super rares, you're talking, you know, 100 nanometers if you're amazing. And the difference is, the gaps between these bacteria are much, much smaller. So we really do need to use electron microscopy. And when we're thinking about these bacteria, it's a bit like thinking about the blind man and the elephant. So we're looking at all these different bits, and I was showing you there's some bumpy bits, there's some bits that look like they might be dividing. So, you know, but what's really going on? And it's like we're looking at different parts of these bacteria and we're not really seeing the whole. So is there a way that we can see the whole? And the answer is, if you've got £1 million, yes. What you can do is you can go and buy one of these microscopes. So this is a serial block face scanning electron microscope, okay? And this is one which is at Oxford Brookes, where I used to be. I'm not at Oxford Brookes anymore, but this is one that's at Oxford Brookes. And just to say, I won't dawdle on this too much, but this SBF SEM was, I had the real privilege to work under Professor Chris Hawes, who was undeniably a titan of plant cell biology. Sadly, he passed away a few years ago. I cannot give a talk as well as Chris about cell biology. He started the cell biology summer school here, okay? And, you know, when he used to come, his knowledge on everything was phenomenal. If you want a taster of that, he has Leaves and Lasers. This is a talk he gave a few years ago at the Natural History Museum. I cannot stress, go and watch it. You are seeing a world-class scientist at his best talking about what he loves, okay? So I really, really would suggest you go and look at that. Anyway, enough of that now. So Chris was responsible for getting this microscope, for setting it up, for getting everything up and running. And what this microscope does, it's a bit like black magic. Like the engineering behind this, I if I hadn't seen it working, I wouldn't believe it worked. So usually you take your thin sections, you get your resin block, you cut thin sections, you put them on grids, you put them in a TEM, you shine electrons through it. This microscope works differently. So what this microscope does is there's a diamond knife in there. Yes, diamond knife in there, okay, very expensive. So there's a diamond knife in there, and what that does is you image the top of the section, so you put the whole block in rather than a thin section, you image the top of it, and you're getting resolution here down to 7 nanometers in XY, so 2 points from each other, more than enough to be able to differentiate between two bacteria. And then this cuts a 100 nanometer. So 100 nanometers, a human hair is about a micron in thickness. So a micron is 1,000 nanometers. This is the last bit of maths I'll give you, okay? And so this is about a tenth of a human hair. It's cutting this section off. And then we're imaging the next layer. So we're imaging sequential sections, one after the other, after the other. And is this going to play? This one doesn't. So what you're seeing here now, so we've taken these we've taken these pea and these beans, and you're seeing these bacteroids, and now what we're doing is we're just sectioning down through the material. We're imaging one after the other, after the other, after the other. Now, I'm selling this a bit short, but this is amazing because you put it in and set it up, and then if it works, that's a very big if. But if it works, you just walk away for a day, and it just sections and sections and sections, and then you worry about the size of hard drive you need to work the data, and then you need to process the data. I also feel really bad. There's a bigger picture of her later, but this is all possible because of Flavia Late. She's the electron microscopist who runs this facility and runs this. I wanted to get these images as big as possible to give you an idea of what we're seeing, but she's absolutely phenomenal. I'll talk a bit more about her at the end. So what you can see, hopefully, as we watch down through these, as we section down through these, we can see that these structures, these almost bifurcated structures coming up here, are really prominent. In fact, most of the cells, when we had a 2D section, it didn't look like that. It only looked like a few, but most of them seem to be. So either there's a hell of a lot of cell division going on, or there's something else going on with the shape here. Whereas this 4292, which appears to have stopped, is much more traditional rod-shaped, stick-shaped bacterioid. Okay, so what we can do now is anyway, had all this data, had tons of data, was struggling for time to analyse it, and then this wonderful thing, I say wonderful, was an absolute disaster happened called COVID, so I had nothing to do, so I sat and analysed data. And when you analyse this data, when you segment this data, what you basically do is you go and you draw in every single Z slice where the bacteria is, and then you reconstruct it based on that, okay? So it takes absolutely ages. And what everyone tells you about artificial intelligence or machine learning, what have you, I'm sure at some point it will work. It does not work yet. I had the experts from the companies that make this software come in, and I was like, here you go, real clear contrast. This is easy. We're going to churn through this data. We'll have it analysed in a week. No, none of the software works. Actually, I won't name the companies, so I'll get in trouble. None of the software worked on it. So so what we can see here, really nicely, I'll go to the next video, which is just we've taken out all the actual EM data, and here we're just seeing the reconstructed bacteria. First of all, we can see, which we could see with the TEM, to be honest, they're very different in size, okay? So they're very different. But what we can see here really nicely is all of these bacteria are actually got this bifurcated shape, okay? So they're all this interdigitated shape, which is really, really, really interesting. And what we can see here also is, so these are smaller, but they almost don't seem to be packed as densely. So that's telling us something quite interesting. This down here is about 100 nanometers, I think, this bar, if I'm correct. But I was asked to give this talk last minute, and I haven't looked that up and checked it, so don't quote me on that, okay? But it might be. I'll find out. So, we go back to the beginning of the talk. Phil asked me, can you tell me the volume of a bacteroid? Yes, I can. So if you can see here in the A34, when we analyse this, so this is a super plot, so we're looking at 120 different segmented bacteria for each one here. If we look at the volume inside the segment, so it's 4.9 times 10 to the 9 nanometers cubed, okay, so proper electron microscopy, nanometers, not micrometers, nanometers cubed, and you see there's a massive difference between the two on the volume inside. That's because in the TEM, even though they did look a relatively similar size, you've got this bifurcated shape in the A34, so they're so the volume inside is much bigger. If we look at the surface area, the volume is also much bigger with the A34. So this is the P species of bacteroid compared to the 4292. And if we look at the surface area to volume ratio, obviously these are much smaller, so their surface area to volume ratio is slightly bigger. And these outliers are interesting, and I think that was a mistake on my part. But anyway, there we go. So, next question is... I told you they were packed. They looked slightly like they were packed slightly differently. So one of the really nice things about SPF SEM data is you get it and you can't just do one, you don't have to do just one type of analysis on it. You can do lots of different types of analysis on it. So what you can see here is, if I play next, is it going to go? And yes, brilliant. So what I've done here is I've taken a fixed volume of the cytoplasm, well, with the bacteroids in it, and I've reconstructed every single bit of bacteria in there. So this is for the A34, and in here I've reconstructed every single bit of bacteroids in the 4292. And this is a lot tighter now. Like I said, this is an old presentation that I pulled up last minute, but these are packed denser. So 47% of the cytosol is filled with bacteroids in P, whereas 40% of the cytosol is filled with bacteria. That's actually dropped now, but I don't want to, at the time when I did this, that was the value that we had of the cytosol is filled with bacteroids with 4292. So, if you think about it, it's a little bit like with that shape and those interdigitated shapes and going together. It's a little bit like when you pack your suitcase, okay? So this is more like my wife when we go on holiday, and that's a bit more like me because I've probably been in the lab up until about an hour before. So, the packing density can be really important and can make a real difference. So, that's basically all I wanted to talk about to show you. So that really is a Gatsby project. That's something that was started here, and over years we've been working on, and we've done here. So Phil asked, can you tell me the volume of a bacteria? And the answer is, with about 6 months and a £1 million microscope, yes, I can tell you the volume of a bacteria. So there we go. So SPF SEM has allowed us to determine differences in bacteroids from P and B. It also allows us to determine the organisation of bacteroids within a nodule. We've done a bit more analysis since then. I really must stress, I am just telling you the microscopy part of this project. Phil has gone and his group have done a hell of a lot more work on fitting a lot of proteomics and flow cytometry, trying to can we get, with this value that I've got on the volume, can we work out the nitrogen fixing capacity of an individual bacteria and that kind of thing. That's where the project's going, but that's all his story. I've just stepped in with the microscopy, okay? So thanks to Phil, his group, Jason and Carmen. Thanks to Oxford Brookes Bioimaging, which is where the imaging was all done when I was a research fellow there, and in particular Flavia Waite. So Flavia's amazing. She's a brilliant electron microscopist, and she kept the machine running and everything. Thank you all for listening. This is from the talk when I was trying to talk someone into us working on symbiosis together, so ignore that bit. But infection threads are very cool, and how they grow doesn't make sense in my opinion, so yeah, we should work on that. And this is because when I was making this talk, I left this in because my 5-year-old son came up to me and said, "Daddy, you're like Batman." And I was like, that's the first time in the world anyone has ever said that. What do you mean? And anyone who's ever seen the Lego movie, Batman only ever my son said, well, all your talks have got black backgrounds, microscopy, black background. And as you know from the Lego Batman movie, black man only wears black or very, very, very dark grey. So that's how your microscopy talk should be. So, different kind of talk. Thank you very much. If anyone has any questions.